So in this next video, we're going to be talking about how to read pedigrees and actually understand the types of pedigrees we can actually encounter. So pedigrees, the general definition is they're a way of analyzing the inheritance patterns of a certain phenotype within a family, right? Pedigrees usually only show a phenotype, and we ourselves have to basically determine genotypes of the certain people in that pedigree or certain individuals in that pedigree. So the basic symbols of pedigrees are circle for a female, square for a male, and all of these are basic. These right here are a little extra, but they're sometimes found in pedigrees, as well as these. These are uh, these are not what you're gonna encounter in a regular genetics class. They're usually for higher level genetics classes. But the main things we have to know is the male, female, and then the unaffected and affected. Affected individuals will mostly be colored in. Unaffected would not be colored in. So to start reading a pedigree, first what we have to do is determine whether a trait is dominant or recessive, right? And that's usually going to be given. If the trait is dominant, one of the parents must have the trait. Dominant traits do not skip a generation. If the trait is recessive, neither parent is required to have the trait since they can be heterozygous. To determine if the chart shows an autosomal or sex-linked trait, X-linked traits are usually linked with males. They're mostly going to be seen in males, but in autosomal traits, meaning traits that are not found on the sex chromosomes, both males and females are equally likely to be affected. So male and females are both going to be affected. Now, tips to read a pedigree. Whenever we're trying to find autosomal traits, it's usually going to be where males and females are both affected. X-linked, either males or females can be affected. Recessive traits usually skip a generation. Dominant traits do not skip a generation. And Y-linked are usually not. We're not gonna really encounter this in our genetics class. So they're usually present in males only, since Y chromosomes are only found in males. So this statement right here says that in X-linked recessive, females will be unaffected mainly, and either will be carriers of that trait or will be normal. Males will mainly be affected or will not be affected. Usually in X-linked recessive, males are the ones being affected. So in an X-linked recessive pedigree, as we're going to see, there's going to be a lot of males that are affected, and that's one of the key characteristics of an X-linked recessive pedigree. So the types of pedigrees and how to identify them. There's going to be three types of pedigrees, main pedigrees you're going to have to know and how to identify. So autosomal dominant, autosomal recessive, and X-linked recessive. So autosomal dominant pedigrees do not skip generations, and in these types of pedigrees, the unshaded individuals are all homozygous recessive, right? So these individuals are all homozygous recessive, so they are not carrying the dominant allele, right? They're carrying both recessive alleles. But shaded individuals can either be heterozygous or homozygous dominant. Since this is an autosomal dominant allele that's not attached to the sex chromosomes, these are non-sex chromosomes. It can affect both male and female, and it's gonna involve the dominant allele, right? So we could either have one dominant allele and one recessive, or we can have both dominant alleles. In autosomal dominant, all we need is one dominant allele to actually make the individual affected, right? To basically make the genotype one that is affected with the autosomal dominant condition. So in autosomal dominant, a key characteristic is that all we need is one allele to make that individual affected, right? So autosomal recessive involves, once again, autosomal chromosomes, which are non-sex chromosomes, so they can affect male and females. So autosomal recessive pedigrees, they can skip generations, but one thing is that they don't always skip generations. There's cases where they do not skip generations. So it's not a set in rule that the autosomal recessive pedigrees, you're gonna see a trend where they're skipping generations. There could be pedigrees where every generation has that disorder, but it's autosomal recessive. So in these kind of pedigrees, shaded individuals are homozygous recessive. Since this is autosomal recessive, we need 2 copies of the allele to actually make a person affected. So this individual has a homozygous recessive genotype. This person as well. And the unshaded individuals that have shaded children must be heterozygous. So these people right here, what it basically says is that these people right here are heterozygous, right? Since this is a normal male times a affected female, this cross made a normal male, and we're bringing in a female, an unaffected female, a normal female, and it made a offspring. They both made offspring that are where there's a 1-to-1 ratio of affected and unaffected. So this is autosomal recessive pedigrees. And then X-linked recessive pedigrees can also skip generations but don't have to. Males only need 1 copy of the gene to show the trait. But if a female has it, all of her sons will have it. So if a female has a copy of an X-linked recessive allele, when she reproduces with another male, in the pedigree it'll show that all of the males will have that certain condition. But in order for a female to get it, the previous generation, her father must also have it, or her mother could have it. But usually it's the father that must have it, right? In order for the next generation female to have the affected condition. So in this case, we see that this male right here is affected, right? So that means that his genotype is X. So let's write A for affected, Y, right? That's his genotype. And he's crossing with he's mating with a unaffected female. It's A, and she could be a carrier, but let's just say she's not a carrier, she's normal, right? So XA, XA. And in this case, when we look at the second generation, we see that all of them are unaffected. So this confirms that these are the actual genotypes of these parents. And if we actually cross these genotypes, you get X. So all of the individuals in this cross are unaffected, right? And that matches up with what the pedigree is saying. This pedigree shows that the second generation, so this is the first generation, parental generation. This is the second F1 generation. They're all unaffected, and this cross is exactly showing that, right? So if you move on to the F2 generation, which is this generation, we see that we see a presence of affected individuals. That means that most of these individuals here are heterozygous, and when they mate with somebody that's being brought into the family, so like this person right here, 7 is brought in, 4 is brought in, 1 is brought in, and when they make their children, we have individuals that are affected. And when you also notice that there's only males being affected. There's no females being affected. And that makes sense since this is an X-linked recessive pedigree, right? It involves an X-linked recessive disorder. And since males only have one X chromosome, see right here, they only have an X chromosome and a Y chromosome. So they only have one X chromosome. So if they have one copy of that gene, that disorder gene, that's enough to actually make that male affected with that certain disorder. So this is X-linked recessive pedigrees. And you can also see that there's skipping generations, right? That's not always gonna happen, but in this case it is happening. So it's another characteristic of X-linked recessive pedigrees. But moving on, we can actually look at some examples now, some actual analysis of these types of pedigrees. So first we're going to look at autosomal dominant pedigrees. So in this first one, the question is, if this pedigree exhibits Huntington's disease, an autosomal dominant trait, what is the genotype of 2-3? So second generation and third person, right? So what is the genotype of that person? So what we want to do is look at the parents and then their offspring, right? So first thing first, right here, the parents show that the male, the father is affected, mother is unaffected. But their offspring, there's a mix of affected and unaffected. And it's not restricted to just males or restricted to just females. It's affecting both males and females, right? And this trait is found in every generation, right? This confirms that this is indeed a autosomal condition, and it's a dominant condition since it's found in every generation. But now, since we know this is autosomal dominant, we know this is going to involve the dominant allele. So whenever we have a dominant allele with a recessive allele, the dominant allele takes precedence. So that means that every affected individual is going to have a dominant allele, right? Whether it be 2 dominant alleles or be 1 dominant allele with a recessive allele. So what does that mean about the unaffected individuals? It means that those unaffected individuals do not have that dominant allele, right? And the only other option we have is homozygous recessive. So all of these unaffected individuals, as long as they're in the family line, they're going to have homozygous recessive, have that homozygous recessive genotype, which involves 2 recessive alleles, right? So let's write, this is a Huntington's disease, so let's write lowercase h. So the unaffected individual is going to have the homozygous recessive alleles. So this is the answer to this first question. Now the second part of the question says, what is the genotype of third generation first person? So third generation first person, we already see that this is a female and she's affected, right? She's female and affected. So the first thing we want to do is look at the previous generation, right? So where did this offspring come from? It came from the marriage of these two, right? From the reproduction of these two. So this is a reproduction of an affected male with a unaffected female. So obviously this unaffected female is going to be RR, right? RR, homozygous recessive, since she has no dominant allele for the Huntington's disease. But over here, he could either be HH. Let me rewrite this. This is lowercase h. It could the male could either be heterozygous dominant or homozygous dominant. But since you see that in the offspring, there's a mix of affected and unaffected individuals. We want to make sure the phenotype the cross actually shows that, and that's that can only be really done when we cross a homozygous recessive genotype with a heterozygous recessive. Heterozygous dominant. So like if we actually do that cross, that's exactly what we're getting. We're getting 50% dominant, 50% homozygous recessive, and the offspring we see that that's exactly where we're seeing a 1-to-1 ratio. One is being affected, one is not being affected. So the only conclusion we can make from this is that the genotype of this individual specifically, third generation 1, is HH. So this is the only applicable type of genotype we can make based off of the parents and the offspring. So one thing about Punnett squares is that there's a lot of things we just can't assume. We just have to basically look at the pedigree and test genotypes. We can't just figure out a genotype just like that with pedigrees. We actually have to look at both the offspring and the parents, and sometimes even the generation before that, to understand the genotypes of a certain generation. So that's one not bad thing about it, but about pedigrees, but one thing that can overcomplicate things in pedigrees. So now moving on, the last question for autosomal dominant pedigree analysis is this one right here. So it's the same question, but is the first generation person number 1 dominant, homozygous dominant, or heterozygous? So we're looking at this individual right here, right? Generation 1, person 1. And we see that first thing we're going to look at is that what's the general genotypes and phenotypes of the offspring. So these two people made this person, this person, this person, this person, and this person. So what we see is a general mix of affected and unaffected individuals. So whenever you see a mix of offspring, a mix of unaffected and affected, it's automatically you don't even need to think about it. It's if you see a mix of unaffected and affected, it's going to be heterozygous. That affected male is a heterozygous dominant male. That means that he has a genotype of A, capital H, lowercase h. Just like Punnett squares, in pedigrees there are certain trends too that you just have to memorize, and it makes the whole process easier. So This is one of those trends. If you see unaffected and affected in a autosomal dominant condition, that genotype of the male parent is probably going to be heterozygous dominant. Now for autosomal recessive pedigrees, they're similar to autosomal dominant. We have a couple questions for that too. So in this pedigree, if this pedigree exhibits Tay-Sachs disease, an autosomal recessive trait, what is the genotype of individual 3 from generation 2? So right here. So when we look at this, since this is an autosomal recessive disorder, autosomal recessive, in order to be an affected individual, we need 2 copies of the recessive allele, right? We can't have a dominant allele, since that dominant allele is going to take precedence over that recessive one and then cancel out, right? Cancel out the effect of the recessive disorder. So we need 2 copies of that recessive allele. And based off of that reasoning, since this is Tay-Sachs, let's use lowercase t, lowercase c. The genotype is simply homozygous recessive, right? We need 2 copies of the affected gene. Now, when we look at the second question, it is saying, what is the genotype of the first person and second of the second generation? So we are looking at this person right here. So for this problem, we're gonna have to look at. We can't just look at the offspring and the current generation. We have to go back to this generation, right? We have to go back to this generation to make sure what their genotypes were, in order to find the genotype of this generation, right? So what we're gonna have to do first, we already see that the female, the mother, is affected, right? And this is an autosomal recessive condition. So what we already proved right here is, in the first question, is that in order to have an autosomal recessive condition, we need 2 copies of the gene, 2 recessive alleles. So this mother is homozygous recessive for the condition, right? And since now that we at least got one of the genotypes, now we look at the second generation, and we see that there's obviously a mix of phenotypes, right? Mix of genotypes, since these 2 people made this person and this person, right? One is affected, one is unaffected. That means that there's a mix of unaffected and affected, and the male is obviously going to have to be a heterozygote to basically not confirm, but in order for this to happen, right? In order for that variation to happen in the next generation. And we can confirm this by actually crossing them, right? So this is the male, female. If we cross this, we get 50% are heterozygous dominant, 50% are homozygous recessive. And based off of the offspring, right, that's exactly what we're seeing. We're seeing one is affected, one is unaffected. And based off of this cross, the affected individual, this individual, is homozygous recessive. The unaffected individual is heterozygous dominant. So based off of this, now we have the genotype of this first person in the second generation, which is heterozygous dominant. Now the last question for autosomal recessive conditions is, what is the probability that the first person of the second generation and the first person and the second person of the second generation would have a 5th child with with this disorder. So we're looking at these two people right here. So these two people, we can already say that this first person is obviously a heterozygote, and this second person is obviously heterozygote, since there's so much variation in the offspring. And she obviously can't be recessive, homozygous recessive, because she's not affected, right? She's not affected. So it's obviously gonna have to be heterozygous parents. Now, if you look at the offspring, there's a mix of affected and unaffected offspring. There's two unaffected right here. I mean, two affected, two unaffected. Now that we have the cross right here, now that we have the genotypes that we needed, we can do a simple cross. Now, what we see in this cross is that there are 3 dominant offspring and 1 recessive. And the question is asking, what's the probability of these individuals having another child with the disorder? And that's 25%. Since 1 out of 4 is homozygous recessive, 25% chance. And that's the answer. Now for X-linked inheritance, we also have problems. So for X-linked inheritance, the question is asking if this pedigree exhibits red-green color blindness, an X-linked recessive disorder, what is the genotype of third generation, second person? So obviously, key characteristics of X-linked is that it usually affects males, and that's what's happening here. 2 males are affected, and it usually skips generation, and that's exactly what happened. This generation does not have it. This generation does. This generation does. So that just confirms that this is an X-linked inherited pedigree. So let's find the genotype of this person. So we know he's a male, right? So he's gonna be X, Y, 1 X chromosome, Y, 1 Y chromosome, and he's an affected individual, and this is a X-linked recessive disorder. So we're gonna have a recessive allele. So the genotype of this individual is this right here. He's affected with a X-linked recessive disorder. The allele is on the X chromosome, and that's the genotype. Now the next question asks, what is the genotype of the second person in the second generation? So this person right here. So first things first, we already know she's a female. So X. Now in order to determine her genotype, let's look at the offspring. The offspring right here are a mix of unaffected and affected. And we already know something about that whenever we see a mix of unaffected and affected, there's usually going to be a heterozygote parent. And it can be the father in this case, since the father was brought in and the father is unaffected. So let's look at the father's genotype. It's obviously XY. The father is unaffected with the X-linked recessive disorder. That means that he has the dominant allele, right? Based off of just logical reasoning. So let's do BB, capital B. That's his genotype. This father's genotype. So he can't be the one carrying the heterozygote. The can't be a carrier, since he just doesn't have the condition, right? But the mother has two X chromosomes, so the mother can be a carrier, and she is a carrier. So this is her genotype right here. On one allele, she's carrying the dominant allele. On one allele, she's carrying the recessive allele. And we basically figured this out by looking at the offspring, right? There's a mix of unaffected and affected. All right, that means there's going to be a heterozygote involved. We look back at the current generation, and we look at what's happening here. So we see that the male is being brought into the family. The male is unaffected. That means that the male does not have that recessive allele. And since males only have an X chromosome and a Y chromosome, and this is a X-linked recessive disorder, that X chromosome can only have that dominant allele. It's not going to have the recessive allele. If that male had the recessive allele, he would have been affected and shaded in, but he's not. So this is his genotype. That basically helps us distinguish which parent has the heterozygote condition, right? And that's going to obviously be the female. That's the only option we have now. So this is the genotype of this person right here. Now the next question asks, could the first person of the second generation, this person, and the second person of the second generation, this person, have a daughter with this disorder? All right, so this question is building off of what we just did. We already figured out the genotypes, right? For the male and for the female. So let's write it down here. So the male right here is XY, capital B. Female is XX, capital B, lowercase b. Now this is just like any other monohybrid cross. So let's do the monohybrid cross. Let's put the male up here, the female right here. Now, just like a monohybrid cross, we just fill out the boxes. So this is what we get as the result of the cross. Now, we're focusing on the daughters, right? Will a daughter have the disorder? So a daughter is a female. So she's gonna have two X chromosomes. So this part of the Punnett script we don't really need right now. It's this part we're looking at. So when you look at the genotypes, you see that one of the daughters is homozygous dominant. One of the daughters is heterozygous dominant. That means that none of the daughters are going to have that condition if these parents are reproducing. So the answer to this question is simply no. They cannot have a daughter with this disorder. And that's based off of, this is basically building off of this question right here. So moving on, those are all the examples for the different types of pedigrees. But what if we're not given that, right? We're not given what type of pedigree it is, and we have to find out what type of pedigree it actually is. So this is an example of that, where you're trying to find the type of inheritance based off of the pedigree. So first things first, what we want to look at is the unaffected individuals and the affected individuals and how they're found in the pedigree. So the first question we would probably ask is, does this does this pedigree skip generations? And that's the answer to that is basically no, right? There's an affected individual in all of these generations. So the first question we would ask is, does pedigree skip? And in this case, no. And since it's not skipping, since it's not skipping generations, this condition is not autosomal dominant. This condition is not autosomal dominant. So this condition is not, and if we go back to what we said about autosomal dominant, this can't skip generations. So actually not. Yeah, so not autosomal dominant, not autosomal dominant. No, so the next condition, the next question actually, is going to be relating to X-linked inheritance. So X-linked inheritance, we already know it's going to affect mainly males. And if any female has at least one of the genes, all of her offspring that are males will be affected. So, So, do any of the females that are carriers have sons with the condition, and all of the sons? So, the only example the only thing, area we see that there's a female and a male mating to make affected males is right here and right here. And we see that these two people make a male that is affected, so we don't need to look at that. But these two individuals make a mix of affected and unaffected, and one of them is one of the affected is a female, one of the affected is a male, and not all the males are affected. That means that this pedigree is not X-linked, right? Not X-linked recessive. So that means that this heterozygous female did not pass on her recessive allele to all the males. She didn't. Now, based off of process of elimination, the only other disorder that's left is autosomal recessive. So this disorder, this pedigree represents one of autosomal recessive condition. And this kind of makes sense because all of the not just males are affected or not just females are affected. There's a mix of males and females being affected. So that means that it's not an X-linked condition, and it's not autosomal dominant and it's definitely not autosomal dominant since over here we already know that one of the females is a heterozygote, so if she was a heterozygote, she would have been affected, right? She would have been affected. But that's not the case here. We're seeing the opposite of that. So she's obviously not autosomal dominant. This is obviously not an autosomal dominant condition. But an autosomal recessive, there's the chance of being a carrier, and that's exactly what's happening here. This female right here has the condition. I mean, not the allele of the condition, and she's a carrier, and she passed it on to two of her offspring. So it's an autosomal recessive condition. So this pedigree overall shows a autosomal recessive condition.